Now I'd like to introduce today's speaker, Dr. Takashi Akera. Takeshi is an Earl Stotman tenure-track investigator at the National Institutes of Health. He is part of the National Heart, Lung, and Blood Institute and leads the Laboratory of Chromosome Dynamics and Evolution. Takeshi received his Ph.D. from the University of Tokyo in biophysics and biochemistry and began pursuing his interest in the cell biology of what is referred to as female meiotic drive as a postdoc in Michael Lamson's laboratory at the University of Pennsylvania. His work has been probative, as you'll see, in illuminating the cellular and biochemical bases for asymmetric chromosome segregation during female gametogenesis in the mouse and leading to drive. Takeshi, thanks for participating in our webinar series and sharing your really interesting work. We really appreciate it. At this point, I'm going to turn the floor over to you.
So today I'll talk about female meiotic drive, trying to understand how selfish elements cheat in female meiosis to preferentially remain in the egg and transmit it to the next generation. So this is a schematic of female meiosis. So female meiosis is inherently asymmetric, producing large egg and small polar body. And you can see in meiosis 1 oocytes, there is a homologous chromosome, a chromosome from dad and chromosome from mom, which is paired to form this meiotic bivalent structure, which will be segregated away in anaphase by this spindle structure, a cell division machinery. And one of the homologue will remain in the egg, and the other one will be extruded out to the polar body for degradation. So actually, polar body is an evolutionary dead end for chromosomes. And according to Mendel's law of segregation, each homologue should have equal chance to remain in the egg. It should be 50-50. But because of this asymmetric cell fate, female meiosis creates a unique opportunity for DNA to cheat this segregation process to preferentially remain in the egg. And indeed, this biased chromosome segregation in female meiosis has been observed in many different species, and it is called female meiotic drive. And because centromeres, which is also a genetic element, direct this chromosome segregation process, they have the best opportunity to cheat this segregation process. And we and other groups have found that expanded centromere satellites show selfishness in female meiosis in mice and also in plants. But actually, there are also selfish elements identified outside centromere region, and we call it non-centromeric selfish elements. Here, I draw them in light blue colors. And so today's talk will cover both selfish centromeres and also how these non-centromeric locus bias their segregation to the egg. And female meiotic drive has been observed in many different species, like in mouse, human, insects, and birds, and also in plants. And it has huge impacts in genetics because it distorts the transmission ratio. And also it has huge impacts on evolution because it changes allele frequencies in the population. And finally, it impacts reproduction because these selfish cheating manipulate gamete production and often leads to infertility. And so here are the big questions in female meiotic drive that we are trying to address. So the first two questions are about selfish centromeres, and the last one is about non-centromeric locus. The first question is about spindle. So there must be some kind of asymmetry between the two sides of the spindle, one side that goes to the polar body and the other side will remain in the egg. And there must be some kind of asymmetry within the meiotic spindle structure. Otherwise, selfish centromere will never be able to find which side will take them to the egg and which side will take them to the polar body. So there must be some kind of spindle asymmetry. And the second question is about centromeres. So what activity makes centromere DNA behave selfishly? There must be something special about selfish centromeres. Otherwise, they cannot bias the segregation. And finally, we want to understand how does non-centromeric locus bias their segregation. These locus are outside centromeres, so usually, as you know, they don't have so much active role in chromosome segregation in general. But still, these non-centromeric self-shell elements can significantly distort the segregation pattern, and we want to understand why and how. So let's start with the first question about spindle asymmetry. So we are using mouse oocytes as a model to study female meiotic drive because we previously established a system to study it. And this is a simple beta tubulin staining to visualize a spindle structure in mouse oocytes. And here we are looking at meiosis 1 oocytes. And you can see that we didn't see any detectable difference between two sides of the spindle, the side that will extrude out the polar body and the side that will remain in the egg. So this tells us that the amount of tubulin is not so different between two sides of the spindle. But this does not preclude the possibility that there might be some asymmetry in post-translation modifications on tubulins, such as tyrosination, glutamylation, and acetylation on these tubulin molecules. And indeed, we found there is a complementary asymmetry in alpha-tubulin tyrosination, with the cortical side enriched in alpha-tubulin tyrosination, and the other side, the egg side, enriched in D-tyrosinated alpha-tubulin. So indeed, we found there is an asymmetry within the meiotic spindle, which could serve as a special cue for selfish centromeres to find which side will take them to the egg. And interestingly, this spindle asymmetry was only observed when the spindle is close to the cell cortex, indicating that cortical proximity is essential to establish the spindle asymmetry. And it is known that cortex overlying the meiotic spindle is polarized and enriched in multiple signaling factors such as CDC42 GTPase and RAC GTPase. So we hypothesize that maybe this polarized cortex is essential to induce spindle asymmetry. So we tested this idea by manipulating these GTPases and found that when we express dominant negative form of the CDC42 GTPase, it diminished the overall tyrosination and canceled spindle asymmetry. And this implies that CDC42 at the polarized cortex can induce spindle asymmetry by increasing tyrosination, specifically near the cortex. So this was good that now we know that there is spindle asymmetry in mouse oocytes and it is regulated by CDC42. But we still don't know if this is relevant to meiotic drive or not. And to test that, we need to establish a system to study meiotic drive of selfish centromeres in mice. So we decided to cross two different mouse strains, one that have larger centromere, and centromere is a repetitive sequence. So larger centromere means that they have more copy number of centromere satellites. And we cross that larger centromere strain to another strain that have smaller centromere, meaning that they have less copy number. And the F1 hybrid is heterozygote for centromere size, and in all sites, they'll have meiotic bivalents that have both larger and smaller centromeres on the same meiotic bivalent. And we can distinguish larger and smaller centromeres in cells using CEMP-B protein, which is a centromere protein that decorates the centromere satellites. And you can see here, the magenta is a chromosome. And here I'm showing two meiotic bivalents. And you can see this is a larger centromere and this is a smaller centromere. And again, this is a larger centromere and smaller centromeres. And we examine if larger centromere preferentially face the egg side of the spindle on the meiotic spindle. And indeed, that was the case. So larger centromere preferentially face the egg side of spindle. So there was a 10% deviation from Mendelian expectation. And now we have a system to study selfish centromeres in mouse oocytes. And next, we want to test if spindle asymmetry is essential for this centromere drive. And we canceled this spindle asymmetry using RAM mutant and CDC42 mutant. RAM acts as an upstream of CDC42, so this RAM mutant also disrupts CDC42 pathway. And under these conditions, the bias was gone, demonstrating that spindle asymmetry in tyrosination is essential for centromere drive. And to summarize this first part of my talk, we found that polarized cortex, especially the CDC42 GTPase, is essential to establish this spindle asymmetry in tyrosination. And this spindle asymmetry is essential for selfish centromeres to preferentially orient towards the egg side of a spindle to segregate to the egg. So I hope I have addressed this first question that I raised in the introduction, but it just raises further questions. And the next question we want to answer, how these selfish centromeres are orienting towards the egg side of spindle. What activity make them selfish to preferentially face the egg side of a spindle? But before getting into the second question, I want to talk about evolution of centromeres. So there is a paradoxical observation that both centromere DNA and centromere proteins are rapidly evolving despite they have essential conserved function in chromosome segregation. So here I'm showing an example of rapid evolution of centromere protein called SEMP-A. You can see here that canonical histone H3 is well conserved across different species. But you can see here that centromeric H3 variant called SEMP-A is quite diverged. And why is that? And 2 decades ago, Steve Henikoff and Harmit Malik proposed this centromere drive theory, where centromere DNA rapidly evolved to cheat in female meiosis to preferentially remain in the egg. But there must be some kind of fitness cost associated to this cheating, which leads to the evolution of the rest of the genome, especially the centromere protein genes, to suppress the meiotic drive and maximize the fitness. And once the cheating is suppressed, centromere DNA will evolve again to cheat in female meiosis, leading to the continuous evolution of centromere DNA and centromere protein genes. So this centromere drive theory nicely explains this paradoxical rapid evolution of centromeres, but it raises basic cell biological questions such as what activity makes centromere DNA selfish, and also how can selfish centromeres be suppressed? So these are the questions that I want to address in the second part. So what do we know so far about selfish centromeres in mice? So we know that centromeres are expanded when they are selfish, and the centromere satellite is called minor satellite in mice. And the centromere expansion leads to recruiting more SEMP-A chromatin, which is the basis of the centromere, as you can see here. And that leads to building bigger kinetochore, which is a protein complex that directly binds to the spindle microtubules. But it was completely mysterious how this building bigger kinetochore leads to their selfish behavior, or in other words, attaching to the egg side of the spindle. So again, we are using this interspecific Mus-Mus hybrid model to study selfish centromeres, crossing larger centromere strain to a smaller centromere strain. And when we carefully live image this hybrid meiotic bivalent, we found that the orientation of larger centromere is initially random. So half of them facing the cortical side of the spindle, and the other half facing the egg side of the spindle. So there is no bias in the beginning. But the attachment between the larger centromere, the selfish one, and the cortical side of spindle was especially unstable, leading to their detachment and flipping of the meiotic bivalent to establish a biased orientation toward the egg side of spindle. And this is the actual example of the bivalent flipping. And again, we are using SEMP-B protein to distinguish larger centromeres and a smaller centromere. And you can see the flipping is initiated by the larger centromere detaching from the spindle first, moving to the opposite side, and the flipping will complete by the smaller centromere moving to the other side. And you can see at this last time point, the meiotic bivalent have the opposite orientation compared to this first time point. And we also found that the directionality of the flipping is biased. So 85% of time, the flipping happens to face this larger centromere toward the egg side of the spindle. And only 50% time it goes to the wrong orientation. So basically this is a source of the bias segregation. Okay, so good. So now we know that there is a detachment and flipping to establish biased orientation, but it is still mysterious. How does this larger centromere, which builds bigger kinetochore, detach microtubule more easily? We initially thought that these larger centromere will hang on more strongly to the spindle because they recruit more kinetochore proteins, but actually they let go microtubule more easily. How is that happening? So we found that larger centromeres not just build bigger kinetochore, but they also recruit more microtubule destabilizers, such as Aura-B and Aura-C kinase, and microtubule depolymerizing kinesin called MCAC. So these microtubule destabilizers functions during mitosis or meiosis to correct erroneous attachment between chromosome and spindle. The chromosome-spindle interaction in cell division is quite erroneous, it's stochastic, so early on there are many misattachments. And these microtubule destabilizers functions to cancel these attachments. And it seems like selfish centromeres are exploiting the same activity to detach from the spindle when they are facing the cortical side so that they can flip to the egg side. So now we know a little bit more about this black box. So selfish centromeres have expanded centromeres DNA, and that leads to building bigger kinetochore and recruiting more microtubule destabilizer to detach from the spindle to flip to the egg pole. But it is still mysterious how does building bigger kinetochore leads to recruitment of more destabilizers, because kinetochore and destabilizers sit on distinct chromatin region, which is centromere and pericentromere, respectively. And we hypothesized that BAB1 kinase is a link which localizes at kinetochore and phosphorate histone H2A at pericentromeres, which recruit shugoshin protein, which is a direct scaffold for these microtubule destabilizers. And to cut long story short, we found that the whole BAB1 pathway was amplified on larger centromeres compared to the smaller centromeres. But still, this is just a correlation. We don't know if this asymmetry in BAB1 pathway and microtubule destabilizer is really important for centromere drive. We want to somehow test this idea. So we decided to cancel this asymmetry in microtubule destabilizer by recruiting BAB1 kinase domain to this pericentromic major satellite region, which is similar between larger and smaller centromeres using a tail construct that directly recognizes major satellite sequence. And in theory, expressing this tail BAB1 construct will increase histone phosphorylation and microtubule destabilizer on both centromeres to equalize their amount to cancel the asymmetry. And this approach worked quite well, and you can see in control case, there is roughly twofold difference in microtubule destabilizers between two centromeres. But when we express this tail BAB1 construct, they nicely localize the centromeres. And then you can see here that the asymmetry was canceled by increasing the destabilizer amount on both centromeres. And you can also see in this quantification here that in control case, there is roughly twofold difference in the destabilizer amount between two centromeres. But when we express this tail BAB1 construct, the ratio becomes closer to 1, meaning that it is getting equalized. And using this approach, we tested if asymmetry microtubule destabilizer is essential for centromer drive. And again, we are using the SEMP protein to distinguish larger and smaller centromeres and examine the orientation on the meiotic spindle. And in the control case, larger centromere preferentially face the egg side of a spindle. But when we expressed Taylor-Babban construct, the bias was gone, demonstrating that asymmetry microtubule destabilizer is essential for centromer drive. And this experiment also tells us that BAB1 kinase is a link between centromer expansion and the enrichment of microtubule destabilizer at the pericentromere region. And finally, we found that centromer drive depends on slowing meiotic progression, so basically delaying anaphase transition. And we found that flipping process takes time. Sometimes this single flipping event takes 1 hour. And as I told you in the beginning, that the orientation of larger centromere is initially random. So you can imagine that if oocyte cells undergo anaphase at this time point, you efficiently suppress centromer drive because you don't give oocytes enough time to flip to the other side. And based on this observation, we want to propose that weak spindle checkpoint in oocytes, which is counterintuitive because of the increased risk of aneuploid egg production, may be actually a consequence of adaptive evolution to suppress selfish centromeres. So reproductive biologists have been wondering why oocyte cells have weak checkpoint, why you want to risk this very important cell division for reproductive success. And here I'm proposing that maybe this weak checkpoint is a consequence of adaptive evolution to suppress selfish behavior because you don't give enough time for them to detach and flip. And to summarize the second part of my talk, we found that larger centromere strain builds bigger kinetochore to recruit more microtubule destabilizers. And this is the activity that make them selfish, make them stronger in female meiosis. And we found that BAB1 kinase is a link between expanded centromere satellite and the recruitment of microtubule destabilizer at the neighboring pericentromere region. So I hope I have convinced you that I have addressed to this first two questions about selfish centromeres. And now we are switching gear to talk about non-centromeric locus. And as I told you in the beginning, these are on chromosome arm region that doesn't usually have active role in chromosome segregation. But still, these non-centromeric locus, non-centromeric meiotic drive locus can distort the segregation pattern significantly. And we want to understand how they are doing that. So this graph shows different meiotic drive loci in different species, and y-axis shows transmission ratio. So if they are, if it is 0.5, they are following Mendel's of segregation. But if it is 1.0, it is completely biased. And strongest one so far identified is called R2D2 locus in mice. And we decided to start from this R2D2 locus. So what is R2D2 locus? So it is a low copy repeat on mouse chromosome 2. It actually sits exactly middle of the chromosome 2. And since centromere is located very close to telomere, this R2D2 locus is really far apart from centromeres. It's 80 megabase apart. And the monomer size of this repeat is gigantic. So it is 120 kilobase. So if you repeat 35 times, you'll gain 4 megabase chunk of DNA in the middle of chromosome 2. And it just encodes one gene called CWC22 within the monomer. It is a spliceosome subunit. And more is better. So if you are heterozygote for this R2D2 locus, expanded one and non-expanded one, this expanded R2D2 shows over 95% transmission ratio distortion. So it's a really strong bias. And there is also a very clear fitness cost to the carrier. So if you're a heterozygote for this R2D2 locus, you have very strong meiotic drive, and there's 30% reduction in litter size due to mild embryonic lethality. So they have very strong drive and very clear fitness scores, but the underlying cell biological mechanisms are completely unknown. And let me also introduce evolutionary history of this R2D2 locus. So ancient mouse chromosome 2 actually didn't have this R2D2 locus, but instead they have this R2D1 locus, which is very similar. It also very similar to R2D2 locus. It also encodes the same gene, CWC22. And roughly 2 to 3 million years ago, this R2D1 locus got copied to neighboring locus and became R2D2 locus. And specifically, this R2D2 locus got amplified over time. And just to let you know, this R2D1 locus itself is conserved from mouse, rabbit to human. So this duplication could have happened in our own genome as well. So we really want to understand this expanded R2D2, how they are cheating in female meiosis. So we first need to establish the system to study it. So we decided to cross the two different mouse strains. One have 0 copy of R2D2, and another one have 33 copy of R2D2. And the F1 offspring is heterozygote for R2D2 locus, and the meiotic oocyte will have this kind of meiotic equivalent. And we really want to understand how this expanded R2D2 locus preferentially segregate into the egg. So we want to label this locus so that we can track their dynamics in female meiosis. But unlike centromeres and telomeres that people have identified so many proteins that you can use to use as a marker, there is no known protein that localized to this R2D2 locus. So we have to first establish a method to visualize this locus. So we decided to use a CRISPR-Cas9 system. It is called dCas9 GFP system. So it is a deactivated version of Cas9 protein. So it still recognizes specific DNA sequence, but it does not induce cutting. And since it has GFP, it will visualize the locus of interest. So we synthesize a bunch of guide RNA that target R2D2 DNA sequence, mixed it with dCas9 GFP protein, and then micro-inject it into oocyte cells, hoping to see these kind of green dot in our cells. And to cut long story short, we eventually found a condition that we can see a nice green dot on one of the chromosomes. And we did similar experiments using a mouse strain that does not have selfish R2D2 expansion. And in these cells, we didn't see these nice green dots, showing that dCas9 is indeed recognizing the expanded R2D2 locus. So now we believe that we have a system to live track this R2D2 locus. And next, we want to understand how they are cheating in female meiosis. And so far, we came up with two possible models for this R2D2 drive model. First one is biased orientation model, which is very similar to selfish centromeres. So in this model, selfish R2D2 locus preferentially face the egg side of spindle on metaphase meiotic spindle, which leads to their segregation to the egg in anaphase. The second model is lagging chromosome model. So imagine that the R2D2 locus can bind to spindle microtubule randomly emanating from the both spindle poles. It will lead to lagging chromosome in anaphase because they are attached to both spindle poles. And lagging chromosome in anaphase may be sufficient to preferentially remain in the egg because cytokinesis is asymmetric in female meiosis, trying to keep most of the cytoplasm in the egg and split a little bit to produce tiny polar body. So even they're lagging exactly in the middle of the spindle, they may have higher chance to be incorporated into the egg cell. And importantly, this model doesn't depend on the orientation of selfish R2D2 in metaphase. Even they are facing the cortical side of the spindle, they can still remain in the egg by lagging. So we wanted to test these models using our dCas9 system. So first, we looked at the orientation of R2D2 locus in metaphase. So we use our DKSNI system to visualize the R2D2 locus, and then examine the orientation on the meiotic spindle. And basically, we didn't see any biased orientation of selfish R2D2 locus towards the egg side of spindle. It was essentially random. And based on this observation, we think it's hard to explain the very strong R2D2 drive by the biased orientation model. And now we are testing the lagging chromosome model by live imaging anaphase cells. So first, we just started by a control experiment, visualizing chromosome by DNA dye in a mouse strain that does not have any expanded R2D2 locus. And you can see chromosomes are nicely aligned in metaphase, and then they segregate in anaphase, and you can see there is no lagging chromosome. And one of the chromosome chunk remain in the egg, and the other chromosome chunk extruded out to the polar body. And in contrast, when we use a mouse strain that is heterozygote for this R2D2 locus, we often see this kind of lagging chromosome in anaphase in many oocytes. And whenever we are able to track the fate of this single lagging chromosome, it eventually joins one of the chromosome mass which remain in the egg. So good, now we know that actually there is a lagging chromosome that preferentially remain in the egg. And we really want to know if this is a chromosome that have R2D2 locus. So we decide to use our DKSNI system to label the R2D2 locus. And indeed, it was the chromosome with R2D2 locus that was lagging in anaphase, while other chromosomes are nicely segregating to the spindle poles. So this data shows us that R2D2 can induce their own chromosomes to lag in anaphase, which will lead to their preferential retention in the egg. So I want to share this working hypothesis. We're still working on it, but we believe that this lagging chromosome is a core feature of this meiotic drive. So let me go through this model. So when R2D2 locus is facing the egg side of a spindle in metaphase, you will produce euploid egg because the other homologous chromosome will be extruded out to the polar body. But when R2D2 locus is facing the cortical side of a spindle, you will have a problem because both homologous chromosomes will remain in the egg, leading to producing aneuploid egg. And aneuploid is lethal for all chromosomes in mice, leading to embryonic lethality. So this simple lagging chromosome explains both the biased transmission and also the associated fitness costs of this drive. So we wanted to next understand how this R2D2 locus is inducing anaphase lagging chromosome. One idea is that maybe they are recruiting centromere and kinetochore proteins so that they can bind to spindle microtubule. So we tested this idea first. So here we are labeling R2D2 locus using oligopane technique, which is a recently developed fish technique that my postdoc, Betsy Clark, implemented into mouse oocyte systems. And you can see here the R2D2 locus is nicely labeled, and she co-stained with centromere protein SEMP-A. And you can see where the R2D2 locus is. There is no SEMP-A that is locals here. And this was same also for kinetochore protein, HEC1. This is a protein that is directly binding to spindle microtubules. And again, you can see that where R2D2 locus is, there is no kinetochore protein observed. So this tells us that R2D2 is not building neocentromere to bind to spindle microtubules to lag in anaphase. So if R2D2 locus is binding to spindle microtubule to lag in anaphase, they must have evolved a completely new way of chromosome-spindle interaction to lag in anaphase. And finally, I want to make a comparison with another non-centromeric meiotic drive loci in maize. So plants have a little bit different female meiosis compared to animals. So they produce 4 equal cells, and the most bottom one become egg cell, or it is called megaspore cells. And maize have this non-centromeric meiotic drive loci called node domain, and node domain recruit kinase motor protein to move faster in anaphase. And moving faster in anaphase is advantageous in their system because it increases their likelihood to be incorporated into this egg cell. So it's interesting that in their system, the node domain system, moving faster in anaphase is advantageous. And in our system, R2D2 system, moving slower seems to be advantageous. So it is like tortoise and the hare, where R2D2 is tortoise and node domain is hare. Either a slower one or faster one can win the race. It just depends on what kind of asymmetry in female meiosis they are exploiting. And to summarize my talk, so we are studying cell biological mechanisms of female meiotic drive using mouse oocytes, and we are focusing on both selfish centromeres and also non-centromeric selfish elements as well. And for selfish centromeres, we found that they preferentially orient toward the egg side of a spindle in metaphase. And this biased orientation depends on asymmetry within the meiotic spindle. And this spindle asymmetry is established through this cortical cue. On the other hand, non-centromeric meiotic drive elements, they don't have biased orientation during metaphase, but they lag in anaphase, which leads to their preferential retention in the egg site because cytokinesis is asymmetric in female meiosis. And with that, I want to thank all the people listed here, especially my postdoc mentor, Michael Lampson, and a very talented undergrad, Emily Trim, who helped my centromere drive work and also initial characterization of R2D2 locus. And now I have my own lab at NIH, and Betsy Clark, a very talented postdoc in my lab, is studying this R2D2 locus to understand how they are cheating in female meiosis. So thank you very much for your attention, and I'm happy to take any questions.
Thanks a lot, Takashi. This is a very lucid talk and really fantastic. Let me encourage the audience to ask questions. We're going to try to do some open mic questions. If you want to raise your hand, we'll try to recognize you to do an open mic question. Alternatively, you can put in questions in the chat, and we'll try to catch those as they come in as well. So let me encourage you to do it. I know there are lots of people out there who have a good understanding of what you were talking about and are working in this field, and I encourage them to ask questions to help the rest of us who are perhaps a little less familiar with all of your work. So there was one question from Hector Kamada, who says, did I misunderstand your original diagram, or would lagging chromosomes lead to aneuploidy?
Yeah, that's a great point. Yeah, so actually that is the key of this model, that this anaphase lagging will not just induce bias segregation, but also induce aneuploidy to kill half of the egg. So we also call it like egg killing model, because basically R2D2 is killing the eggs that are trying to extrude them out to the polar body and keep them alive when they are trying to segregate the R2D2 locus to the egg. So if this theory is all true, half of the egg will die, which will reduce the litter size, which is observed. And the other half, the survived one, will most likely have this R2D2 locus. And this is also consistent with the observed very strong bias, like nearly 100% will have this R2D2 locus in their offspring. So this model, is consistent with the observed strong bias and the fitness cost. And this lagging chromosome does induce aneuploidy as well. Yeah. Thank you, Hector, for the question.
Well, thanks for the answer. Actually, I posted that question about 5 seconds before you actually talked about it in your talk, so it was a bit premature.
Yeah,
but your elaboration on that point was much appreciated. Thanks.
Thank you.
I'm going to ask a basic cell biology question. If you could elaborate a little bit more on a weak spindle checkpoint in oocytes, And could you just talk about that checkpoint in oocytes that you're referring to?
Oh yeah, sorry, yeah, that's a great point. So this is, their cells have many different kind of checkpoints, like DNA replication checkpoint, and also the spindle checkpoint is basically a checkpoint that monitors if chromosomes are properly attached to the spindle apparatus. And if there is like some misattachment of chromosome, then cells can arrest the cells in metaphase so that they will give more time for the proper chromosome spindle attachment to prevent errors in anaphase leading to aneuploidy. And if checkpoint is weak, if spindle checkpoint is weaker, then cells doesn't wait even chromosomes are misattached, leading to missegregation. And oocytes in general have weaker spindle checkpoint, like this has been observed in different species, like human, pig, and also mouse and xenophas. Like xenophas eggs have literally almost like no detectable checkpoint. They just undergo anaphase instantly. So it's a conserved feature of female meiosis that they have weak checkpoint, and people just wondering why you want to risk this very important cell division. And now we are thinking maybe it is a strategy to suppress selfish behavior. But this is still like a speculation. We need more data to support this, but at least it is consistent with our observation so far.
Yeah, great, thanks. There's a question from Pratima Tranuri, and she says, "Thank you for a great talk and a naive question." Don't worry about the naivety of the questions; they're all good. I just asked one. So, if meiotic drive were sex-linked, does meiotic sex chromosome inactivation have a role to play?
Interesting. Yeah, that's a great point. Yeah. So far, there is no a known link between. To my knowledge, misregulation in sex chromosome inactivation and the female meiotic drive, but actually X chromosome itself is known to also show meiotic drive if they are univalent. So this is actually interesting. So in mouse XO, so if you have X chromosome, just one copy of X chromosome, but not the other one. Actually, XO karyotype become female. So you will undergo female meiosis. And under this condition, it has been shown that this univalent X chromosome preferentially remain in the egg. And the molecular mechanism behind that is still unclear, but we are thinking maybe that could be explained by this lagging chromosome model that I showed. Univalent chromosomes are shown to be misattached with a spindle and often lag in anaphase. And we think this is not just R2D2, but any kind of chromosome. We think if they lag, they can preferentially retain in the egg. So we think this meiotic drive of univalent X chromosome could be also explained by our lagging chromosome model. But sorry, as far as I know, I don't know any link between sex chromosome inactivation and female meiotic drive.
Yeah. Good. Keep asking questions, please, from the audience. Jeremy Hollis has a question. Says, "Great talk," and it was, if you were a slow polar body, if you were a slow polar body extrusion, do you imagine you could rescue the drive? If you could slow polar body extrusion, do you imagine you could rescue the drive? And the reference here is to the R2D2 system, I think.
Yeah, that's a great point, because I think, like abscission, the cytokinase completion, if that is too fast, then maybe, it's not just giving enough time for the lagging chromosome to be incorporated into the polar body. So yeah, that's a great point. And that's something we haven't tested, but I think that is totally possible that maybe, cytokinase is just shutting down the gate too early so that lagging chromosome cannot be segregated to the right pole. So yeah, that's something we could try. And yeah, maybe we can treat with low dosage of actin drug so that we, slow down the constriction, or, myosin inhibitor. Yeah, that's something interesting to test. And we are also thinking to test maybe the size of the polar body matters, because the size of the polar body is also slightly different between different strains or species. And when we make hybrid, probably due to some kind of hybrid incompatibility, the polar body size also changes. So one possibility is that when the polar body is bigger, then you reduce the chance to retain R2D2 in the egg. Hope I'm clear, but so basically the division should be like highly asymmetric to preferentially keep the R2D2 locus into the egg. And if you make this less asymmetric, like making polar body bigger, we can imagine that the R2D2 drive will become weaker. So kind of rescuing the drive. So that's something we also want to look into it. Yeah, thanks for the question, Jeremy.
Good. Sarah Zanders has a talk. Hi, Sarah. Great talk. Do you think the mechanism of R2D2 lagging is similar to lagging of chromosomes without homologs?
Yeah, that's a great point. Yeah, so lagging chromosome without homologs is typically mediated by centromeres. So centromeres are in meiosis 1, they have sister centromeres, but they are stick together, so they behave as one unit. And if they have only one unit of centromere, they cannot be properly bioriented, and they just keep misattachment, correcting misattachment, leading to lagging chromosome. So this is a centromere mediated lagging chromosome. And the R2D2 lagging chromosome, it could be similar. So at least we know that they are not building centromeres, but it could be building some other, we think it is recruiting some other microtubule binding protein, but we think it doesn't have to build something really sophisticated like kinetochores, maybe just recruiting some microtubule stabilizing protein that will lead to some lateral attachment of microtubule. And even their lateral attachment, they should be still able to induce anaphase lagging. So the mechanism could be slightly different, but we think that it is similar in the sense that they are both recruiting microtubule binding protein and then leading to anaphase lagging. Hope I understand answer to the question.
Good. Well, if Sarah, feel free to follow up as for anybody else as well. Katie, I'm going to say something to a person who helped me out on the back end here. Katie, I can't see any hands raised with my side. If there are any, just let me know, okay? Because I guess I'm not seeing any of that. I don't know if folks are raising hands or not. Let me go to another question. This question is from Siddharth Sivanandan. Does the movement of the R2D2 loci lag with the entire chromosome, or could this occur as a mini chromosome or truncated chromosome piece? With the lack of centromere proteins seen, I would think a truncation is more likely. I'd be very interested to hear your views on the same. And you could see this question, Takashi, if you didn't pick up the question, you can probably see it in your chat.
I have that chat box open, so I can see it. Yeah. So this is also a great question. So we sometimes actually see in anaphase or telophase that there is some like small chromosome fragment that harbor R2D2 locus. So basically, we think they were under tension when they are pulled by microtubule in anaphase, so that sometimes they just break and then split, separate from the rest of the chromosome. So this first indicates that they are under tension. They are probably pulled by microtubule during anaphase. And second, they could actually behave just by itself without centromere. So we are really eager to test if like a mini chromosome just harboring R2D2 locus can behave as selfishly as this R2D2 locus on mouse chromosome 2. So we have some plan to make like a mini chromosome and see how they behave on mouse chromosome, or maybe we can do something a little bit more classic, but a little bit crude, that we can do some laser ablation so that we, basically ablate the centromeres of chromosome 2 so that it will be just a chromosome chunk with R2D2 without centromeres. And we can see if they are still behaving similarly to lagging anaphase and preferentially remain in the egg.
Great. Okay. Let's see. Another question here from Andreas Huben. Andreas, thanks for coming today. And just note that I'll make two notes. One, Sarah Zander will be one of our speakers in this webinar series later on, and so will Andreas. And Andreas has a question related to B chromosomes. Excellent talk. R2D2 behaves like a non-disjunction region of the Ry B chromosome. In both systems, it's the combination of asymmetric and chromosome lagging non-disjunction. So just I guess a point of similarity that's being noted.
Yeah, that's a great point. Yeah, from my next talk, I'll have that together with maize node domain. Yeah, you're right. That's maybe more appropriate comparison. Yeah, that's a great point. Yeah, thank you, Andreas. Yeah.
I mean, I know Andreas is going to talk about B chromosomes in a couple of weeks, but and maybe I'll just ask this question that's more general. I mean, the mechanisms that you've were illuminating today, are they seen in other systems? I mean, B chromosomes certainly are a system, and we see that there's some similarities. But can you just comment on other mechanisms?
Yeah. So I think this idea that lagging chromosome is causing female meiotic drive is new. I don't think there is any other system that has been shown that lagging chromosome is a driver of female meiotic drive. But there are like B chromosome, they don't have, they don't really pair in meiosis, so they are behaving as univalent, and univalent they typically lag in anaphase. So I don't think it has been proposed to my knowledge, but it's possible that this B chromosome that is not paired are lagging in anaphase, and that also contributing them to preferentially remain in the egg. The system that I know is the grasshopper systems, that they have asymmetry in the spindle structure. So one side, the egg side, have 3 times longer spindles compared to the cortical side. And B chromosomes just randomly locate on the spindle, and they just have higher chance to be captured by the egg side of the spindle because they are 3 times bigger. And that leads to their segregation to the egg. But I think also in this case, the lagging chromosome should be happening in anaphase that could further increase their likelihood to be getting into the egg.
Great, thanks a lot. And let's see. Brandon Fagan has a question here. Also says, great talk. You mentioned that mice may have evolved a weak spindle checkpoint in order to suppress centromeric drive. Why do you think this is worth it? What are the consequences of centromeric drive? All right, so good question. All right, go ahead and speculate.
Really great question. Yeah, so basically, so definitely. So if there is no downside, no fitness cost for centromere drive, you don't have to risk female meiosis like this, like making checkpoint weaker. But and first of all, I have to say that the fitness cost of centromere drive is still unclear in different systems, but all of the systems that we use have fertility issues. So we are still trying to address if this fertility issues is directly coming from this selfish behavior, or it's something indirect. But that is something encouraging, that maybe the selfish behavior or selfish centromeres itself, even they're homozygous, can lead to some fitness cost to the host. And so based on our finding that selfish centromeres, once they're expanded, having more microtubule destabilizers, we can think about possible fitness costs from this finding. Because if you can imagine, if you have too much microtubule destabilizer, then you never be able to establish that stable attachment with a spindle. So basically, this will also lead to missegregation. So you can imagine if you make centromere bigger and bigger, making selfish and selfish, actually it will risk both mitosis and meiosis because you cannot attach to the spindle properly anymore. So again, we are still, investigating what is the fitness cost of having selfish centromeres and centromere drive, but we think there's some missegregation involved in that, and that is the driver that is driving for the rest of the genome to create some suppressor mutation, and we think that could be a weak checkpoint. Yeah, but yeah, this is still just a speculation at this point, and thank you for the question.
Yeah, good. Great. I'm going to ask a question of my own here that we're coming to the end of the chatted questions, and I still recommend people, if we have some more, good, that's good, keep them coming. I do want to ask, again, sort of a naive question, but something, just a clarification really, and it had to do with the R2D2 chromosome lagging, and I didn't quite understand how that was occurring. What was, were you getting attachments at both spindles? And could you just describe that?
Sure, yeah. So I'm still sharing, right? So
yes, you are.
So I'll just go back to my model where I have my spindle. So the idea we have is that this R2D2 locus acquires some activity that they can bind to spindle microtubules from both spindle poles. And when it's like a tag and war. So if you're attached to both sides, it will be it will be pulled by both ends and never be able to simply pull to one of the poles. And so that will lead to lagging chromosome in anaphase. And one way is that they recruit microtubule binding protein so that they can attach to these spindle microtubules. But actually, I want to actually keep it open because we haven't actually shown that R2D2 locus directly bind to spindle microtubule. Actually, it could be some other ways that they can lag in anaphase. And there are many different structures like around the spindle region. And maybe R2D2 is just binding to some other structure, hanging on to some kind of structure that does not change their position during anaphase. And that should be also sufficient to lag in anaphase. Like microtubule are segregating other chromosomes, and R2D2 chromosome, they just hang on to that structure so that they maintain their position. So it could be some other structure like actin meshwork or some like ER membranes, mitochondria. There are many structures surrounding the spindle. So I still want to keep it open that maybe R2D2 is exploiting other structure surrounding the spindle area.
Yeah, okay, good. Sarah Gilmore says, amazing talk. Is there a minimum size of the R2D2 repeats for the drive system to be effective? And or does the effectiveness of the drive scale with the size of the array?
Yeah, that's a great point. So at least it has been shown that so the system we are using have 33 copy of R2D2, and it shows like 95% transmission ratio distortion. And the discoverer of this drive, Fernando Pardomenia de Villena, have shown that if you get down to 11 copy of R2D2, then it's completely Mendelian. So it completely drive is suppressed. And I believe they also tested like 20 copy of R2D2 and it was intermediate. So the copy number matches with the strength of the drive. But actually, having said that, they also found that there must be some secondary loci somewhere in the genome that mediates the strength of this drive. Because even you have this high copy R2D2, if you cross to certain strain, then that meiotic drive become much, much weaker. So there must be some modifier locus, which is not identified yet, which is controlling the strength of this drive. So yeah, there are some different elements contributing to the strength of the drive.
Okay, great. Thank you, Pratima, for asking another question. Down here it says, does the mouse R2D2 have anything to do with Star Wars or the Drosophila R2D2, which forms the RISC complex in the RNA pathway? I was thinking the same thing. Good question. Thank you for asking that.
Yeah, thank you. So I know at least it's not relevant to the Drosophila R2D2. That's an R2D2 gene. And actually, I should have clarified that. But this R2D2 is not the name of the gene, but it's a name of the chromosome region, which contains a gene and some like repetitive elements. And R2D2 stands for responder to drive number 2. So, responder is a locus that experience bias transmission. So the discoverer, I didn't ask him personally, but yes, I'm not sure, maybe he's a big fan of Star Wars, but it's not me that named this. And I don't know why, but R2D2 is an abbreviation of responder to drive number 2. Yeah.
Great. Super. Okay. Well, I think our audience has basically run out of questions, and I just want to let me actually, before I'm going to grab the screen from you, and